Good afternoon, my name is Helena and I am going to present the impact of Botrytis cinerea on wine aroma of the Portuguese cultivar Trinket. Trinket is a Portuguese grape variety commonly used in dry red blend wines from the regions of Alentejo, Dão and Douro, although in this area it is called Tinta Amarela. It has an average size cluster with compact berries of a dark blue color. However, it is highly susceptible to Botrytis cinerea infection and has also a very irregular growth depending on weather, which make the wine aroma quite inconsistent throughout the years. Wine aroma is dependent on volatile organic compounds. There are many families that contribute differently to the aroma. There are terpenes, sugar and amino acid derivatives, fatty acid derivatives, and they can be in their free volatile form or as a glycosylated form. To approach Trincadeira aroma, we harvest 4 healthy clusters and 7 infected by Botrytis cinerea, and we separate the work in 2 parts. The first was the volatile identification and quantification of both free and glycosylated volatiles using beta-glycosidase, and we ran them on the GCMS and GC feed. The second part of the work was to see the gene expression of genes involved in volatiles metabolic pathways. So we extracted RNA and we did some qPCRs targeting these genes based on the literature. As a first result, I show you an OPLS of the volatiles, and we can clearly see the control samples and the infected samples being separated by different groups of volatiles. In this case, we have ester fatty acids contributing for the MOX side, and on this side we have ethyl estates contributing for the infected separation. As this OPLS gave such interesting results, we further went to see how each volatile was changing in the infection. We are using as a unity a volatile percentage of total volatile content. And the first problem we saw was that ethanol was really high and it was masking others' volatiles percentage. So we separate this in different graphs that we cannot cross compare, but we can look at them individually. So in this case, we have, for example, amyl alcohol, ethyl acetate, and acetic acid that are showing presence only in infected samples. On this plot, that is the free volatile plot, we can see N-hexanol increasing and 2-transhexanol decreasing, and these two are both part of the LOX pathway. Regarding the glycosylated volatiles, the first thing we see is that we have many more volatiles here. And it's very interesting to look at the ester acetate family, as they are only present upon infection, all of them. It is also quite interesting to notice that ester fatty acid, ethyl canoate, and ethyl dot canoate are in very high concentration in mock samples, but upon infection, they both decrease. So this was the first part. Now, regarding the gene expression, we tested 15 genes that were coding for enzymes and proteins involved in metabolic pathways of the volatiles, both free and glycosylated. And we only have one change in HPL2 that is increasing upon infection. So HPL2 is involved in the LOX pathway as well. Unfortunately, none of the other genes change, so I guess it was the wrong time point to check this expression. So in summary, what we can see is all the volatiles that changed upon infection, and we can see here the fatty acid esters all decreasing, and the show up of the ester acetate family only in infection. We also have the appearance of decanoic acid and acetic acid, which are not what we expect in a wine flavor, a good wine at least, that show up only in the infection. And we have like the lipoxygenase pathway changing, as we saw as well. So these volatiles can and might be used as positive and negative markers of infection to check for the quality of the grape or wine. So in summary, 35 volatiles were detected and 19 were statistically different between infected and control. Infection is leading to the activation of the LOX pathway with the increase of the linoleic acid, that is a precursor, and the N-hexanol, plus the upregulation of the HPL gene that we confirmed. We didn't detect terpenes or others, and most of the volatiles were present in their glycosylated form. New volatiles, mostly from the ester family, were detected upon infection, and they might have an impact on grape and wine quality. I would like to thank everyone on the team that worked on this project and also thank to the conference. Feel free to ask any questions. Thank you.